Right, good morning everybody. Very early morning. Sorry about the delay, but there was some confusion in the schedule. I'm surprised that everybody's here because my schedule set a completely different room, but anyway. Right, you're now finishing a course where you were taught about signaling between cells, the different kinds of signaling, and now we're starting the last sort of mini block that's going to be about signaling in the nervous tissue between neurons mainly. So today we're going to go through a bit of an overview of the general mechanisms of signaling between nerve cells and within nerve cells. And later on so next week in the later lectures, we're going to be talking about specific neurotransmitters, their chemistry, their receptors, and we're going to talk a little bit about pharmacology of these receptors. So what kind of drugs we can use to influence the signaling in the nerve tissue. Right. So how is, especially if we talk about the central nervous system, but also in the peripheral nervous system, how are signals transmitted? What are the different kinds, what are the different modalities of signaling, if that makes any sense? Yep.
Through neurons?
Sorry?
Through neurons?
Yes. So within neurons and between neurons, what kinds of signals can be transmitted?
Synaptic signaling?
Okay, and what kind of signaling is that? Okay. Right, so there is electrical signaling and there's chemical signaling, right? Is that correct? Those are the two modalities that I was looking for, okay? They're quite distinct, but both of them are present in the nervous tissue. There's electrical and there's chemical. In most of the lectures, we're going to be talking about the chemical modality, so neurotransmitters, their receptors, etc. But let's start with the electrical signaling. So where does that occur? Does it occur within cells or between cells? Yes?
Within the cell, depolarization of the membrane causes electric impulse.
Very good. So the main place where electrical signaling takes place is within cells, within neurons mostly. But not only, there's also electrical signaling between cells, and I'm going to mention it a little bit when we talk about the synapse, because there are also electrical synapses. But let's leave that for later. Okay? So let's now talk about how this electrical signaling takes place within a neuron. So if we draw a sort of a prototypical neuron looking something like this, what is this? Okay, so that's the body of the neuron, or soma. We have a nucleus there, and some other stuff. What are these things? Dendrites, very good. What is this? It's an axon, good. So within this nerve cell, we need to transmit a signal from dendrites into the axon. Obviously, you know from histology, or you will know, that there are many different kinds of neurons that look different to this, but the general gist of it is the same. Right, so we need to transfer the signal from the dendrites to the axon and to the synapse at the end, and then we're going to talk about the synapse. And we said that the mode of transmission of the signal is electrical. And you mentioned that it's about depolarization. So do you want to explain, or someone else, do you want to explain what does it mean, what kind of depolarization, what is being depolarized, and what does it mean? Whoever wants to. Yeah. Okay, which membrane is it that's being depolarized?
The axon membrane on the axon?
Okay, so
you first have to let some, for example, calcium in to get that small.
okay.
And then you can open the voltage to the and start.
Okay, so it's clear that you know most of the way. Yeah, did you want to?
Yeah, the membrane of the target cell is depolarized by inputs of sodium ions.
Okay.
There's a change in membrane potential.
Okay. Right, so the answer to my question, which membrane is depolarized? Well, it's the cell membrane. Why do I insist on making that clear? Because there are other membranes in the cell that also have membrane potentials, such as mitochondrial potential, okay? Which is not the I mean, that's not what we're talking about, okay? So it's the cell membrane, it's the cytoplasmic membrane. I know it's trivial, but I think it's important to mention that, okay? So the cell membrane of the neuron is depolarized. Now, the suggestions that I got was that sodium ions play a role, calcium ions potentially play a role there. There was something about the axon, that the depolarization starts in the axon. So let's try to figure the whole thing out. So where does the depolarization start? Yeah?
Dendrites.
Okay, it starts on the dendrites, one or more of them, because that's where the axons of other neurons end. Okay? This so far I guess is trivial for you. Okay? So the axons of other neurons terminate on dendrites, usually on dendrites of the next cell, of the target cell. And obviously, these synapses release neurotransmitters, and we're going to be talking about that quite a lot. There are some receptors there, and they cause the activation of those receptors cause depolarization. Now, well, they don't have to. They can also cause hyperpolarization, depending on what receptors we have, but let's assume that it's depolarization because we want to activate, we want to transmit some active signal. Right, so there was somebody mentioned that depolarization starts on the axon. Can somebody elaborate on that? Yeah?
Sometimes it starts from the axon, but just goes there. Instead of going one direction, it goes in both directions. Like the electrical signal itself, instead of going in one direction,
okay.
it goes in both directions.
Yeah.
It starts in the axon.
Okay, yeah, that's a very unusual situation, but it can happen, I agree. Is there something special about the axon as opposed to the dendrites and the cell body?
Myelin sheets.
Hmm?
The myelin.
Sure, but some axons do have myelin sheets, some don't. About the type of depolarization, is there something special about the kind of depolarization that occurs in the axon and the kind that occurs in dendrites? Yeah?
Dendrites are negative gated, axons is voltage gated.
Not quite. There's some truth to it, but yeah.
Do the axons stimulate action potentials, and sometimes they can jump across from cells and not just through the cell.
Okay, so the word or the term that I wanted to hear is action potential. So depolarization is a general set of possibilities. What does depolarization mean, by the way? What does it mean, just generally? What do we mean by depolarization? You don't have to raise your hand, just shout.
Changing action potential.
Decreasing action.
Yeah.
Also, difference in charge.
Decrease the difference.
Mine is
Okay, so it is a change in the resting potential. It's not the action potential, okay? Not yet anyway. So it's a change in the resting potential of the membrane, and usually, because what is the normal resting potential of a cell, of a general cell? What is the range?
60 to 70.
Okay, 60 to 80-ish is for neurons. But for general cells in the human body, let's say it's between 40 and 85 or something like that. Okay? Those are the usual potentials that you find. For neurons, it's between 60, 70, thereabouts. Now, is it plus 60, minus 60? And what does it mean if we say it's minus 60 or plus 60?
It's minus 60 inside the membrane, plus 60, I think, outside the membrane, because inside is negative.
Okay.
Probably more negative than outside.
Okay, so it's, let's say, minus 60 or minus 70, and that means there's negative inside. Okay? Important thing, because it's important for the flow of ions. Now, before we get to the actual depolarization, how is the resting potential set up? What actually creates, what causes membrane potential, in this case specifically? Yeah?
Potassium. That potassium can have these free gates that they can flow through, like through inside and outside.
Okay, how does that set up the potential?
Because it's more the calcium inside and outside. So when you have these free gates, the calcium.
Is it calcium or?
Oh, yes, sorry.
Okay, potassium, yeah.
Because it's higher concentration inside, probability for it to go out is higher,
Mm-hmm.
and that creates more minus inside, and that connect, that doesn't go inside again because it wants to connect to the anions inside. So that creates the potential.
Okay, raise your hand if that's clear to everybody. Okay. Okay, right, so I'll just repeat it for those of you who are hearing that for the first time, which is fine because, you know, that's what the lecture is for. Right, so what your colleague said was that the basis of a resting potential is a difference between ion concentrations inside the cell and outside the cell. The two main ions that have different concentrations between the outside and the inside are sodium and potassium, okay? What are the differences where there is more sodium and there's more potassium? Yeah.
More potassium inside, more sodium outside.
Okay. Do you know approximately the concentrations? It's probably a good idea to have some idea about those concentrations. Yeah.
145 millimole of potassium inside the cell and 120 millimole natrium outside.
Okay, that
and
wasn't...
outside is potassium 4.5 millimole, and inside is natrium 12.5.
Okay, so it wasn't quite right, but you were almost there. Okay, so outside...
No, it was correct. Pretty sure.
Okay, well, you might be pretty sure. But outside we have about 140 of sodium, approximately millimolar. Okay, about 140 millimolar, depending obviously on the person. Of sodium. Okay, about 140 millimolar-ish outside, okay? And inside, it's between 10 to 20 millimolar, okay? Depending on the cell, it's different in different cells, okay? Now, potassium, that was absolutely correct, so it's around 4 millimolar. Potassium outside is about 4 millimolar, and it's about 100 millimolar-ish inside, okay? So it's not exactly symmetrical, but those are approximate values. But it will obviously differ between different cells because it's not exactly the same for all of them. So anyway, those are the basic differences in the ion composition. But that on its own would not be enough to create resting potential, okay? But as you correctly said, at rest, a normal cytoplasmic membrane of a cell tends to be permeable to potassium, but not to sodium, okay? So the difference in concentrations, the difference in the concentration of sodium is really irrelevant because sodium can't really move anywhere, okay? The membrane is impermeable to sodium. But if it is permeable to potassium, based on the concentration gradient, potassium is going to try to leave the cell and go outside. And as you said, it's really just about probability, okay? So yesterday when I was teaching the Czech students, they were asking, well, you know, why don't, when we talk about the ion channels, they were like, well, there's this huge pressure of those ions trying to push into the channel. Surely they would destroy the channel or something like that. But there's no such pressure. The ions don't really know that there are more of them inside. Okay? Just imagine that they are randomly, you know, moving around and hitting the ion channel, which is basically a hole through a membrane. And sometimes they go through and sometimes they don't go through. But obviously, if there are more of them inside, the probability that they will go outside is much higher than the other way around. Does that make sense? Just raise your hand if that makes sense. Yeah? Because sometimes people really think that the ions somehow are pushing out or something like that, or pushing in. It's not really like that. It's just random movement, and the probabilities are like that. Right, so if potassium is trying to get out of the cell, because there's more of them there, well, it's not trying to. I guess I'm actually making it worse when I say those things. Okay, so it tends to go out more than it goes in. Because it's positively charged, it obviously takes a positive charge with it, which with each ion, the inside is becoming less positive, it's become more negative. Okay, when does that stop? When does the process of potassium leaving the cell, when does it stop? Or how can we stop it? There are several different ways to stop it. Yeah.
By equilibrium? By equilibrium? If it's
Okay, when is equilibrium reached? In this case. So we have potassium ions, certain concentration of potassium ions in, certain concentration out, and the membrane is permeable to those ions. So when is equilibrium reached?
Are you looking for the number?
No, just describe the state. What happens?
When you achieve resting potential.
It's not quite, well, can you explain that? Yeah.
Because I assume that resting potential is the potential, the membrane potential at which you have an equilibrium between the ions inside and the ions outside. So that's when you actually know that potassium is not going to move anywhere, or sodium is not going to move anywhere.
Yes, you're right, but I think the explanation is not quite there. Yeah?
When inside the cell is more negative than outside the cell?
So even when it's -1 millivolts, it's going to stop?
No.
Okay. Okay.
Does the probability is the same?
Well, when does that happen? When the probability is the same?
Equilibrium, I suppose. Oh, if the concentrations are the same.
Okay, so one way to stop the efflux of potassium ions from a cell is to equilibrate the concentrations, to have the same concentration on both sides, right? Okay? If that happens, the ions have no need to move. Well, they will still move, but the probabilities are going to be the same, right? But that actually never happens in our body, because it's unlikely, it's highly unlikely, and actually would kill you, if your extracellular fluid had 100 millimolar potassium or something like that. So that never happens. But there's something about the electrical potential. So the equilibrium that is reached is not really an equilibrium of concentrations. That's never going to happen. Okay? But there are two forces, sort of imaginary forces, acting against each other. One is the concentration gradient, which pushes potassium ions out. And the other one is the electric field, which is pushing all positive ions, but the only ones that can move through are potassium ions, for the membrane is impermeable to all the other ones. So there are two forces acting against each other. Okay, the gradient pushes potassium ions out, and the electric field pushes them in. And when these two forces, which are not really forces, well, the electric force is a real force, but anyway, when these two tendencies are the same, then the movement will stop. The overall movement will stop. Okay, does it make sense? Yep.
But what about the sodium?
Okay, at rest, the membrane is impermeable to sodium. So the sodium just sits there.
Even if it's the electrical force that pushes it?
It can't go through. There are no channels that are open for sodium. So there's nothing it can do. It might feel the push, it probably does, but it can't go through. So there's no net movement of sodium. Okay? Now, this equilibrium between the two forces is reached not really at a resting potential, at least not in most cells, and I'll explain that, but it's reached at what we call an equilibrium potential for potassium, okay, which is approximately -90 or -95 millivolts. So it's actually a lot more negative than what we find in normal cells, okay? So equilibrium potential for potassium. Now, the question is, why do we have resting potentials that are much lower than that? Well, the reason is that it's not only potassium that causes the movement and that causes the setting up of the resting potential. There are other ions, there's chloride, there is calcium, there are anions, protein anions inside the cell, and these all interact to set the exact number for the resting potential. Okay? So the main reason for it is potassium, but it's definitely not the only one. Otherwise, if it was just on potassium, the resting membrane potential would be the equilibrium potential for potassium, which is about -90 or 95. Okay? So this is how the resting potential is set up, and we agreed that for a neuron, we're somewhere between -60 and -70-ish millivolts, negative inside. Right, let's go back to depolarization. So depolarization means changing this relatively negative, well, it's definitely negative, but relatively highly negative potential. It's actually a difference of potentials, it doesn't matter. Potential towards more positive values. Okay, that's depolarization. Making it more positive means the membrane is depolarized. Yeah.
Just one thing, everybody's talking about that the equilibrium potential of potassium is, like you said, -19, of natrium a little bit lower, like -70, but nobody says why some two positive ions can have a negative potential. I mean, potassium is K+. How can equilibrium potential be -90 millivolts?
Well, that's what I just tried to explain. The equilibrium potential is basically just a state where the tendency of potassium to move out of the cell is balanced by its tendency to move in based on the electric field. Okay, and this situation occurs at approximately minus 90 because there's so much more potassium in. Okay, if the situation were different, the potential would be different as well. Okay, so this is for the specific situation in our cells. Right, so depolarization occurs on the dendrites, just general depolarization. The membrane potential becomes more positive. But we mentioned one very important term, which is action potential. Action potential is also a type of depolarization, but it's a very specific type of depolarization. Okay, action potential is a depolarization that is highly coordinated. It uses very specific ion channels to actually occur, and it moves quite nicely unidirectionally along the axon. Now, action potential, and I'm going to mention a bit more about it, only starts in the axon. Okay? Now, those of you who are really interested in neuroscience will find that there's some debate whether some kind of action potentials might be occurring in the dendrites, but for now, let's assume that action potentials only start in the axon. Okay? So what happens in the dendrites and in the soma, in the nerve cell body, is just a normal depolarization. So what happens there, if this is the potential difference, and this is time, what we see starting, let's say, from minus 60, the potential just becomes more positive and stays there, and then it goes back or something like that. Okay? Just a simple depolarization, a simple change in potential. Now, once this depolarization reaches the axon, something completely different happens. And what we see there is that as the depolarization reaches, let's say here, reaches the threshold value, which also is different for different things, a very dramatic depolarization occurs, and then a little bit slower repolarization. Okay, and I'm going to explain what happens in those processes. Okay, so you can see that the time course of the depolarization is very, very different. Okay, so this is the action potential. It's also a depolarization, but it's a very specific kind of depolarization. Okay, this is just a general kind of depolarization. This is action potential. Okay? Now, how is action potential mediated? I'm not going to talk now about how this general depolarization is mediated, because we're going to talk about it quite a bit when we talk about individual neurotransmitters and their receptors and what they do to the cell. So I'm going to talk about the molecular basis of action potential now. Okay? Now, we said that at rest, the plasma membrane of a neuron in most cells is only permeable to potassium, okay, not to sodium. But what happens here, and what is the cause of this massive and pretty quick depolarization, is that sodium channels suddenly open. Okay? And as sodium channels open, because there's so much more sodium outside than inside, what happens to sodium ions? They go in. And since they are positively charged, obviously they're going to make the inside potential more positive, and therefore what we see is, so if this is minus 60, we can say that here is whatever, 10 millivolts or something. Okay, plus 10, thereabouts. Okay? So the potential becomes more positive. Sometimes it becomes actually positive, so there is a reversal of potentials. Okay? It doesn't have to happen, but it often happens. Yep? Okay. Right, so I mentioned sodium channels that are suddenly open. Now, what opens them? Okay? Unlike on the dendrites, where the usual stimulus for the opening of various kinds of ion channels or what have you are the neurotransmitters, here the stimulus is the depolarization itself. Okay? So this relatively small depolarization that just moves passively along the dendrite, here it causes the opening of a special kind of sodium channels, which are called voltage-dependent or voltage-gated sodium channels. Remember when we had a lecture on the muscle, we talked about voltage-dependent and voltage-gated calcium channels. Okay? These sodium channels are very similar. So now I'm going to talk about the molecular structure, and you can be sure that basically all voltage-gated channels, all voltage-gated ion channels have very similar structure and very similar mechanism of working. Yes?
Do you also find them on the cell body, or are they only?
That's one of those things that is being debated by neuroscientists. There are probably some, there are almost definitely some voltage-gated sodium channels on the nerve body and on the dendrites, but the density is not sufficient to actually support this nice action potential. Okay?
It's sufficient to propagate the signal and
They're definitely sufficient to propagate the signal, but not in this form. Okay, so, right. So let's now have a look at the voltage-gated sodium channels, because I want you to understand their molecular structure and how they actually work. Right. Now let's draw a schematic view of a sodium channel. So this is a voltage-dependent sodium channel. And obviously at a resting potential, it is closed. So there is some kind of a domain that closes the channel, okay? So that sodium ions cannot go in, okay? The channel is closed. This is resting potential, let's say minus 60 millivolts. So there are a lot of negative charges inside, a lot of positive charges outside. Okay, so this is the resting state of a voltage-gated sodium channel, right? Now, what we want is for the channel to open when the potential changes. When the potential becomes less negative inside, or even positive inside, it doesn't really matter, just the potential changes. Okay. Now, how would you design a protein that would respond to changes of membrane potential? Now, those of you who've already watched the lectures or whatever, try not to answer, not to spoil it to everyone else. Okay. How would you design a protein that would react to the electric field, to the changing electric field in the membrane or on the membrane, across the membrane? How would you do that? Okay, have you all watched the videos? You have? Okay. Well, why am I even lecturing?
Maybe by having some charge.
Okay. Where?
The gate. So.
Okay. So what would happen when the potential changes?
It can be drawn to some of the charges.
Okay, and that's exactly how evolution developed all voltage-dependent ion channels. Basically, there is a domain, or actually several domains in the channel that have positively charged residues, amino acid residues. What would those be? What amino acid residues can be positively charged, or will be positively charged at normal pH physiologically? Indeed, the basic ones. So,
Histidine, lysine, and arginine.
very good. Okay, histidine, lysine, and arginine are the ones that could be expected. Now, histidine is a bit of a tricky one because there the protonation occurs exactly around the physiological pH, so it can change quite a bit. But lysine and arginine are definitely going to be positively charged under normal pH. Okay, so very good. What we have in all those voltage-dependent ion channels, there's a positively charged domain that, as you said, responds to the electric field. So in the resting potential, when it's very much positive outside and very much positive, negative inside, the domain is pushed down by the electric field. Okay, it's being repelled by those positive charges being attracted by the negative charges here. Now, when the potential changes, that electric force changes as well, and the domain moves up because suddenly the repulsion and the attraction are smaller. Okay? And that opens the channel so that the ions can go in. Now, this is a schematic drawing. It doesn't actually look like that at all. The channel is a sort of a cylinder, and the voltage sensing domains there are 4 of them, and they're around the cylinder. And what happens is that when the potential changes, they move up and they rotate. And by the movement and rotation, they open the channel. And then when we'll see during repolarization, then rotate back and close the channel. Okay, so it doesn't actually look like that, but this is the mechanism of it. Okay, right. So this is what happens during some depolarization. It doesn't have to be huge depolarization. It's actually relatively small. Suddenly, those sodium channels, voltage-dependent sodium channels, open and allow the influx of sodium inside the cell, which causes a massive depolarization, actually potential reversal in many cases.
Does it happen in the dendrites or in the axon?
Now we're talking about the action potential, so we're talking about the axon. But as we just discussed with your colleague, these ion channels can be found on dendrites and the body as well, but they do not, at least in most neurons, they do not support an action potential. Okay, so they may open, they may propagate the depolarization, but they will not cause something like that because the density is not high enough. For an action potential, you actually need a sufficient density to propagate very quickly and to cause these kind of depolarizations. Okay, right. So we opened the sodium channels and caused huge influx of positively charged ions in. But then, obviously, we need to return the neuron to the original state. So we need to repolarize the membrane. How are we going to do that?
Close the
Okay, so first we probably want to close the sodium channels, okay? Because otherwise they would just keep letting sodium go in, which is not something that we want. So we need to close them. How are we going to close them?
Taking out the sodium, pumping the sodium out of the cell.
That does occur indeed. Sodium is actually throughout all the time it's being pumped out. But first we need to close the channels because there's no point trying to pump out the sodium that goes in because we would be just wasting lots of ATP and we wouldn't be doing anything. Okay? So, I mean, maybe there are other ways, but I think the first thing we should do is to close the channels. Now, how do we close them?
Automatically close. Automatically close.
Right. So the first idea that everyone would have, well, obviously we just reverse the potential and the ion channel closes. Okay, obviously, and this is actually what does happen in the end. But the problem is, how do we put the potential back where we started? Okay, we're here, we're at plus 10 or plus 20 or whatever. You know, there's no way to reverse the potential, and there's still lots of sodium ions coming in and flooding the cell. Well, actually not that many, but anyway, substantial numbers of sodium channels going in. So the reversing the potential is not really a viable option. We would need some other hugely expensive mechanism to reverse the potential so that these ions close. So as your colleague said correctly, there is actually another domain in the protein that automatically closes after a while. Okay, approximately after 1 millisecond, 1 or 2 milliseconds. Okay. So we start with depolarization. The voltage sensing domain moves up, opens the channel, and after a few milliseconds, another domain just automatically snaps shut and closes the channel.
What kind of domain is that? Is that also in the amino acids kind of thing? Or is it...
Oh, absolutely. It's part of the protein. Yeah, it's just another part of the whole channel. It's not charged because it doesn't have to be. The stimulus for the closing is not a change. Well, it is a change in the potential, but it's actually this change in the potential. So the closing of this inactivation gate, okay, because this state of the channel is called inactivated. This is closed, okay. Then it's open. Sorry, I actually drew two states in one go, which might be confusing. So this is open state. And then a few milliseconds later, we get the inactivated state. Okay? So closed, open, inactivated. And the stimulus for the inactivation comes from the opening. So the movement of the voltage sensing domains causes a slow movement, well, relatively slow movement, of the inactivation gate that just closes after a few milliseconds. Okay? So 3 different states of the voltage-gated sodium channels, and actually this is true for many other voltage-gated channels as well, for calcium channels, for potassium channels. Many of them also inactivate after a few milliseconds. Okay? Yeah, that was a question.
You said that in reality it's 4 of them and they just go up and down.
Yeah.
So is it a different like this automatic thing? Is it different than the four proteins?
Yes, it's a different part of the protein that inactivates it. It's actually, well, I can send you the papers. It's sort of a very strange domain that looks like that. It's called a ball and tether hypothesis. So it really looks like a small ball of protein that just after a while moves in and blocks the pore. But it's a different domain than the 4 voltage sensing, but the stimulus comes from the voltage sensing.
Well, they're still up, this closes it, and then they come down.
Well, we'll get that. But yes, indeed. Are there other questions? Yep.
It's just the movement of the opening of the gate that actually signifies for the third domain to close.
Correct. Only there's a slight time delay of a few milliseconds, but correct, yes. Okay? Yeah?
How many milliseconds did it take before it closed the other domain?
What, this domain? About 1 or 2 milliseconds. Okay? Right, so the sodium channel now is inactivated, so effectively closed. It's just a different state, it's inactivated. No more sodium channels can flow in, so that's sorted. But we still are somewhere here, because just closing sodium channels is not going to return the potential back to where we started. Okay? So we need some other mechanism to take it back where it was. How do we do that?
Pumps, sodium pumps.
Okay, sodium pumps, they do play a very tiny role, but pumping of sodium is, first of all, very expensive. It uses a lot of ATP, but more importantly, it's very slow. We don't actually have that many pumps on the membrane because we're talking about, 100 milliseconds or something like that, probably less for most neurons, 20 milliseconds. There's just not enough sodium pumps to do that in such a short period of time. Yes?
Is it because the next one on the axon opens? The voltage-gated ion channel opens?
It does, but how would that return the potential back in?
You pump potassium out, release.
Okay. So what happens, and it happens almost at the same time as sodium channel open, potassium channels open as well. Okay, so there's just a very slight delay, so we can say that here sodium channels open in just a few fractions of a second, potassium channels open as well. There are also voltage-gated potassium channels, so the mechanism of their activation is exactly the same. They also respond to the depolarization, and they just open. Okay. Now, so the depolarization of the membrane opens both voltage-dependent sodium channels and voltage-dependent potassium channels. Both of them open. Okay. Potassium channels are slightly delayed. Their opening is a bit later. Okay. So basically, most of the potassium influx occurs when the sodium channels are already inactivated. Okay, so the sequence of things are slight depolarization of the membrane, it reaches threshold, sodium channels, voltage-dependent sodium channels open, okay? Now, potassium channels open, sodium channels inactivate, and the potassium channels just keep working, they just keep being open. Does that make sense? Can somebody explain how that repolarizes the membrane? Yep. Anybody? Yeah.
Since we have a bigger concentration of potassium inside than outside, if we still keep, if we introduce more, if we put more sodium ions inside the membrane, we are going to make it positive. But if we close the gates and then start taking out the excess of potassium from inside of the cell, we're going to actually reduce the action, the membrane potential.
Okay, so exactly like that. We closed the influx, we stopped the influx of sodium inside. So the potential is not getting any more positive. That's sorted. But since we already opened potassium channels, the potassium, and there's still way more potassium in than out, the potassium will start to flat out. Okay, will start to move out. Okay, and since it is positive and it's moving out, it's going to start moving the potential back to negative values. And in theory, it will go all the way down to -90, which is the equilibrium potential, but in real life, it doesn't really, for various reasons. Okay, so it goes back to, let's say, -60, where we wanted it. And the whole thing basically returns to normal.
So that graph is only countable for one voltage or one channel?
Yes, this graph is actually only valid if this is the axon. So we're just looking for one slice of the axon. Okay? And the whole thing very quickly moves to the next slide, to the next slide, and this is how the action potential is moving along the axon. Okay, so it depolarizes here, then it depolarizes here, it starts to repolarize here, then it depolarizes here, here it's already repolarized, and it moves like that. Okay? So the return of the inactivated state back to the closed state, which can all open, because this one we can't really open it, okay? We have to put it back to the closed state and that one can open, is achieved by returning the potential back to normal resting potential. Because as the voltage sensing domain moves back to its original place, it also releases the inactivation gate.
But if the membrane then is at resting potential, how does that affect the next one? If the resting potential now is achieved again,
yep.
What makes the next one open?
Well, but this takes much longer than stimulating the next bit.
So when the next one is opening, that one hasn't closed it?
Very likely, yeah. Yes. Okay? So this whole process takes, let's say, 50 milliseconds, depends on the type of neuron. Okay? It takes a very long time. So by the time this has repolarized, we're already somewhere here with the depolarization. Okay? Just 1 second. Very important thing and very important function of the inactivation. Well, one of the reasons why we have it, one of the reasons is so that we can repolarize afterwards. But the other reason is that it actually doesn't allow the signal to flow backwards. Okay? Because by the time these channels open and depolarize the area around them, these channels are inactivated. So they can't open again. Okay? So this basically achieves that the depolarization, the action potential, only moves unidirectionally. Okay? And that's also a significant difference between the action potential and just general depolarization. General depolarization can spread in any direction it wants. Okay? That's why I said that action potential is highly coordinated. It's actually a very specific type of depolarization. And one of the features of it is that it's unidirectional. It always moves in one direction. Okay? If you start it the normal way. Of course, if you electrically depolarize the axon somewhere in the middle, of course, the action potential is going to spread in both directions because there's no reason for it not to. But if it starts in the beginning of the axon, it always flows in one direction.
So it's originally depolarization of the membrane from the dendrites that makes all the voltage-gated channels along the axon.
Correct.
Yeah.
Correct. Yes?
How can the potassium be stored back inside the
Right. Yes. A very good question. And it was kind of hinted on by other people. Obviously, by the end of this whole process, even though the channel is back in the original state, or all the channels are back in the original state, we still have a slightly different concentrations of ions in and out, because we moved some sodium in and we moved some potassium out. So we also need to return those concentrations back to the normal, otherwise we wouldn't be able to repeat this whole cycle. And for that, we have the ion pumps. Mostly the sodium-potassium pump, which you all probably know, the NAK pump. Yep. Okay. So that's one of the major ATPases, major pumps that works to restore the ion concentrations. Now, probably an important thing to say is that the movement of ions that is necessary for this huge change of potential is actually very tiny. Okay? So during this action potential, the actual number of ions that move in and out is very, very small. We're talking about like millions of ions or something like that, which, considering the scale, is nothing. Okay? So what I'm just trying to say, during a normally functioning of a nerve cell or muscle cell, the actual movements of ions are very, very small. So if you put some kind of a very special device that would measure like ion selective electrode or something that would measure the changes of ion concentrations in the middle of the cell, you wouldn't see any. There's just no bulk changes of concentration. It all happens around the membrane. Okay? The differences are tiny and they only occur around the membrane. Inside the cell, inside the axon, you wouldn't really see many changes. Okay? This is just to put it into perspective. So the ATPases are very important, but actually they move relatively low amounts of ions if everything is working properly. Do you have a question? No. Right. Okay, we're going a little more slowly than I thought. So let's have a 5-minute break, and we're going to continue after that. No questions? Yep.
So the mechanism for the opening of the inactivation gate, to put it back to normal, is it the second gate that just changes, it does the conformational change and then it opens it, or how does that work?
The opening of the inactivation gate is basically just a reversal of its closing here.
Which goes first?
Of which
Does
events?
the inactivation gate open first, or is it the...
No. First, the voltage sensing domain has to go back to its original state, which opens the inactivation gate. Okay? Any other question? Yeah?
How long does it take to go from signaling until it's ready to receive another signal?
Yeah, that's a good question. I don't actually know the number. It's going to be tens or hundreds of milliseconds. I don't actually know. I think it also depends on the specific nerve fiber, because different nerve fibers have different refractory periods, but I don't actually know that number. But there is a substantial delay. You can't just send signals one after another, very short, separated by very short periods of time, because they would be just lost. Because they would hit the axon when it's still inactivated, and there would be nothing going on.
Does it mean that if you're like burning yourself, you won't feel anything for some like milliseconds because you can't like
well, one thing is the delay between your finger and your brain.
Yeah.
So that obviously is there because it takes a while for it to travel. But this is more about the separation of different signals. So imagine that you would have a pin and you would want to prick your finger. Okay, it's a bit of a stupid because it wouldn't work. But imagine that you would be trying to prick your finger and the pricks would be separated by 1 millisecond. Can't do that, but imagine that you could. Okay, the second prick, you wouldn't feel it. Okay, you probably wouldn't feel it because your brain can't actually distinguish such short periods of time. But what I'm trying to say, even the nerve wouldn't actually register the second prick. It wouldn't carry it because at the time when you would do the second prick, it's in the inactivated state, so it couldn't register it. Okay, it's a bit of a stupid example, but sort of. Okay. Yep.
I was a little bit unsure about what the function of the potassium was.
Right. Since based on the ion concentration differences, while sodium, when sodium channels open, is going to try to go in and it's going to make the potential more positive. If you open a potassium channel, potassium is going to go out and it's going to make the potential more negative. So it's going to return the potential back to the original value. Okay. Right, let's now move on to the synapse. So from electrical signaling, we're going to move to chemical signaling. Now, I said that I'm going to tell you about electrical synapses. So very briefly, while in, at least numerically, most synapses in the human body and in most mammals' bodies, I guess, most synapses are chemical. Okay, so we're basically going to be talking only about chemical synapses. But there do exist electrical synapses, which are basically just gap junctions between two neurons. So the depolarization or the action potential coming from the first neuron just traverses the gap junctions to the second neuron, and that's how an electrical synapse works. Does it make sense? Yeah?
Would an electrical synapse function quicker or faster than a...
Yes, it is. Yeah, they're definitely faster. I think there's some research that shows that there are some electrical synapses in the retina, in the human retina, but clearly at least their abundance is tiny compared to chemical synapses. So we know, and also their functioning is so simple that there's no need to circle them. Just remember that there are gap junctions between different neurons. Okay? Now let's talk about the normal type of synapse, the chemical synapse. So if this is the presynaptic and postsynaptic neuron,
just one thing. Isn't it in the heart the same that we have those gap chunks?
Absolutely, yeah. Yes, in the myocardium you have a similar mechanism, but since there are no nerve cells, we don't call it a synapse. Okay, so the intercalated discs, that's a sort of a similar thing as in electrical synapse, but it's not a synapse because it's in a muscle. Okay, right. In a prototypical chemical synapse, there are lots of different things, both inside and outside the cells. So can you tell me some structures that we find in the presynaptic, well, actually anywhere. Just tell me where they are. What can we find in the cells or outside the cells in a synapse? Okay, so there's some neurotransmitter vesicles. Yep. The mitochondria. What are the mitochondria for? Sure, yeah, they produce ATP for lots of things.
Calcium channels.
Calcium channels. Okay, calcium channels.
Proteins.
Proteins. Well, I guess that's all one big bunch of proteins, but anything else?
Receptors.
Okay, what kind of receptors and where are they?
I'm pretty sure they found both of them on the pre and post.
Okay, so we have postsynaptic receptors, which are responsible for transmitting the signal into the next neuron, which is, well, to the dendrite usually. Okay, and there are also presynaptic receptors, on the presynaptic neuron. What is the function of those? What do you reckon?
Some kind of feedback.
Very good. Their function is basically to give some feedback information to the presynaptic neuron on how much neurotransmitter has been released into the synapse. Okay? So very often those presynaptic neurons are inhibitory because we want a negative feedback. Okay? The more neurotransmitter is being released, the more we want to limit further release of the neurotransmitter. Okay? So just remember, presynaptic neurons usually inhibitory, and they provide negative feedback. Yep.
Will the neurotransmitters also be transported back into the cell?
Okay, very good. So there are some neurotransmitter transporters for the reuptake. We're not going to talk about them today very much, but yes?
Does the feedback of the neurotransmitter or how much neurotransmitter you send have anything to do with how strong you feel the sensation?
Yes. That's about as far as I can talk about it now. But yes, absolutely. Yeah. They are very important, and they're important in treatment of certain disorders, and we're going to talk about those.
Voltage-gated calcium channel on the slide.
Yeah, I actually drew the voltage-gated calcium channel right here, and I'm going to explain in a minute why I drew it there. Because most of them are actually right next to the vesicles, because that's where they're needed. Okay, there are definitely some here as well. Sure, I can draw one there as well. Anything else? Do we have ribosomes there? Yeah. Okay, yeah, sure.
No, we don't have any ribosomes.
No ribosomes? Okay, good. Yeah, correct. No ribosomes. So all the proteins are synthesized in the body, okay, fairly important, and then they are transported along microtubules and some other cytoskeletal structures. Okay, lots of other things there. I'll just mention that there are some extracellular enzymes, and we talked about one of those extracellular enzymes in the synapse that are quite important for, yep.
Acetylcholinesterase?
Yeah, remember we talked about acetylcholinesterase. That's actually an extracellular enzyme. It's secreted by the neuron, so it's outside, and obviously its function is to get rid of acetylcholine to stop the signaling.
Is it free in the synaptic cleft, or is it?
Well, it's actually bound to the postsynaptic membrane somewhere here. It's not just floating around, but it's attached. There are obviously a lot of extracellular matrix proteins and lots of various adhesion molecules and stuff like that, but you know, we would probably be here for a fortnight if we just wanted to mention all of them. Right. I'll just mention one other structure which is quite important for the functioning of the synapse, and I wonder if anybody knows what it is. Very good. It's a process, a projection of an astrocyte. So what I just want to stress here, astrocyte, that more and more research now shows that synapses and signaling especially in the central nervous system, is not only dependent on neurons, so it doesn't need just neurons, but it also needs glial cells, and specifically astrocytes. Now, in histology, you probably had pictures, nice pictures of astrocytes, and you saw that they are hugely branched, and they have lots of different projections into various areas of the central nervous system. And usually, those projections, they attach to a synapse, and they actually play a very important role in the functioning of the synapse. One important role that they play is an uptake of neurotransmitters. So we said that there are some uptake proteins, pumps, transporters in the presynaptic terminal where we can re-uptake the neurotransmitter. But also a lot of neurotransmitters are actually taken up by the astrocyte. Okay? Now the other important thing, and they are then recycled, and we're going to talk about that. We're going to talk about them when we talk about the specific neurotransmitters. But the other important thing is that astrocytes can also transport signals. They can transmit signals from one place to another, because they also have receptors on their membrane for the neurotransmitter. So the release of a neurotransmitter into the synapse also activates the astrocyte. Now, astrocytes don't actually have action potentials. Okay, they can't do action potentials. But what they do instead is they have waves of calcium that move through the astrocytes. So the activation of these neurotransmitter receptors, for example, activates calcium channels. There are lots of different ways of doing that. And this increased concentration of calcium then moves as a wave, as a sort of action potential, but it's not really, it just moves through the astrocyte, and it can influence another part of the astrocyte which is attached to another synapse. And it can then influence by secreting or transporting or doing all sorts of other things, can influence other neurons. So this is just to stress that even though all the attention has been paid to neurons mostly, glial cells and especially astrocytes are probably very important, not just for sustaining all the functioning of the neurons, but also for information processing and the proper functioning of the brain. Now you know that there are about 10 times as many glial cells as there are neurons, so you can imagine how important this whole thing, which is still quite poorly understood, is. Yep.
Does the astrocyte send its waste products to the microglia?
In general, no, but of course microglia as immune cells, they do mop up lots of various proteins and dead cells, etc. But in general, I mean, at least in these processes, it's not really important.
If they can recycle neurotransmitters, can they also secrete them?
Yes, they can.
Okay, but wouldn't that mess up this?
Well, you can call it mess it up, or you can call it just is part of this of the whole thing. Okay, the same way as a neuron can secrete it, astrocytes can secrete it. They secrete it in a different way, and not all of them can do it. And it's, as I said, those are processes that are quite poorly understood so far, but they definitely can secrete some of them, yes. Especially the amino acid ones. There was another question there? No. Right. In the last part of the lecture, which looks quite a bit shorter than I would like, but anyway, I'm going to talk about the molecular mechanism of neurotransmitter exocytosis. How these vesicles are actually released, what is the stimulus, what are the proteins involved. Okay, so let's have a close-up schematic of the presynaptic membrane with one vesicle containing a neurotransmitter. Now, the neurotransmitter, whatever it is transported into the vesicle using specific transporters. Okay, actually, most of the time, those are the same transporters that then re-uptake the neurotransmitter from the synapse. But anyway, so the neurotransmitter goes in, and what we need to do is to take the vesicle and fuse it with the target membrane, with the presynaptic membrane, so that the vesicle opens. So this is the synapse, this is the synaptic cleft. Okay, and we need to fuse this vesicle with the target membrane. Right. In order to do that, there's a huge number of proteins that are needed. It's actually a very complicated process, endocytosis as a whole, but we're going to talk at least about some of them. And structurally, and functionally, the most important proteins are known by an acronym, SNARE. Okay, it's really an acronym. I don't want to tell you what it stands for because it's quite complicated, but anyway. So that's why it's written in capitals, okay? It's an acronym. So these SNARE proteins are attached both to the vesicle and to the target membrane, which in this case is the presynaptic, the cytoplasmic membrane. Do you all understand where we are in the cell? Okay? This is obviously massively out of scale, but maybe it's going to make it a little clearer. Okay? So this is the presynaptic terminal. Yep.
Are the ions who are pumped, or neurotransmitters who are pumped into the vesicles?
Yes, they are. Because you have to fill the vesicle with the neurotransmitter. The neurotransmitter is synthesized somewhere else, usually in the cytoplasm, depends. And you have to pump it in to a sufficiently high concentration first before we do. Yes.
Oh, I thought it was...
No, only peptide neurotransmitters, only proteins are synthesized in the body. Okay? All the other neurotransmitters are synthesized in the synapse itself. Either synthesized or recycled, depends on what's going on. Okay? Right, so we have those SNARE proteins there, and we can distinguish two kinds of SNARE proteins. One are called V-SNARE, standing for vesicle, and the other group are called T-SNARE, which stands for target membrane. Okay? Why such a stupid name? Why not just call it cytoplasmic, C-SNARE, or whatever, cytoplasmic membrane, or something, or plasma membrane, P-SNARE? The reason is that there could be different kind of target membranes. For example, in the endoplasmic reticulum, when vesicles coming from the endoplasmic reticulum want to fuse with Golgi apparatus, it uses very similar protein machinery. So therefore, the target membrane can be other membrane, doesn't have to be plasma membrane. Okay? So that's just for the nomenclature. Okay? Now, those SNARE proteins contain a specific domain, a motif, a part of the protein, which is called a SNARE motif. And this is this helix that I drew there. And the interesting property of these helices is that they really want to stick together. Okay? They're like Velcro. They just really want to, once they're close to each other, they just stick together with huge force. Okay? And this is exactly the mechanism that we use in exocytosis to get the membrane of the vesicle very, very close to the membrane, to the cytoplasmic membrane, so that the two actually can fuse and the vesicle can open. Okay? Now, in addition to these two yet unnamed SNARE proteins, there's another one there, which actually has two SNARE domains. I'm going to tell you the names in a minute. Okay. So we have 3 proteins in total. but actually 4 snare domains, the domains that like to stick together, okay? Because one of them has 2. Does it make sense? Yeah? Okay. So we have, in total, we have 4 snare proteins that just sit there next to each other. Yeah?
But is the V snare and the T snare like come together?
Yes.
Or is the other one as well?
So we have 2 T snares, this one, and we have 1 V snare, and they all come together.
Oh, so the red one also is a T snare?
Yes, because it's on the target membrane. Okay, I'm going to tell you the name so that we don't have to speak in riddles. This one is called SNAP25. This one is called syntaxin. And this one is called synaptobrevin.
Where is the SNAP25 attached?
Right. Both syntaxin and synaptobrevin are integral membrane proteins. So they go through the membrane and they're attached as a protein in the membrane. SNAP25 is not an integral protein. It's only attached to the cytoplasmic membrane via a palmitic acid residue. So it has a fatty acid residue that attaches it to the membrane because the fatty acid likes to be in the membrane. Okay? So it's not an integral protein. It doesn't have actually, this is not a protein chain that attaches it. It's just palmitic acid. Okay? It's actually a common feature in many enzymes that, enzymes or proteins that need to be attached to membranes, that you put highly hydrophobic stuff on them. Okay? I think you talked about the RAS protein. RS, yep, okay. It's attached to the protein from the to the membrane from the inside. Did you talk about how it's attached? Okay, there is a myristic acid residue, again a fatty acid residue that attaches it. So it's a very common feature. So SNAP25 is attached just using palmitate. All the other ones are integral membrane proteins. They actually go through the membrane. Yep.
So the V snare is the
synaptobrevin is a V snare. There are other ones. Okay. Syntaxin and SNAP25 are T snares, but there are also other ones. Okay. So T snare and V snare are the broad category. Okay. Those are the specific types that I want to talk about. Right. So now we know what they're called. We know that we have in total 4 snare domains, and what happens is that they just snap together like Velcro, 2 pieces of Velcro, and that pushes the vesicle towards the membrane. And obviously, once that happens, we get something like that. The 2 membranes fuse together, a neurotransmitter can go out, and we basically just have one membrane with a nicely tangled complexes of snare proteins, like that.
How do they get attracted to snap together?
Yeah, that's what I'm going to tell you now. Okay, so this is the actual molecular mechanism of fusion, or at least the initiation of fusion. There are obviously some steps that are still not entirely understood, but this is the main point of it. But we only want the vesicles to fuse with the membrane when a stimulus came to the synapse and the synapse is told, release the neurotransmitter now. We just don't want it happening all the time, right? The stimulus for that, as in many other cells, is an increase of calcium concentration. So when the action potential comes down through the axon and it goes through the cycle that we just talked about, depolarization with sodium channels, etc., etc., the depolarization of the membrane in the action potential also opens voltage-dependent calcium channels. Okay, so calcium channels are open, and again, I'm going to draw one here, and they allow calcium to go in. Once again, I drew the channel right next to the vesicle because this is in fact where they are. Remember when I told you in the action potential, the number of ions that actually move around is relatively small. And one of the reasons for the whole setup, so that the numbers of ions are small, is so that the cells don't have to spend a lot of energy trying to pump them out. Same thing is here. The calcium channel is right next to the place where it needs to be, so that we only need a few ions of calcium to go in for the whole thing to start. If the calcium channel were here, we would need a lot of calcium ions to move around so that at least one of them gets to where it needs to be. Does that make sense? Right. So we have calcium going in, and now what we need is a calcium sensor. So we need a protein that senses the concentration of calcium, and that actually starts this whole process. And the protein is on the vesicle, and it's called synaptotagmin. 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 T-A-G-M-I-N. Okay, synaptotagmin. So that's the sensor. It's a protein that binds calcium. Once it binds calcium, it changes its conformation, and the change in conformation basically brings together the snare proteins and allows them to form the snare complex and causes the fusion. Now, I'm leaving out approximately 40 other proteins that play a role there, okay? So this is a massive simplification, but it tells you the most important, the core processes that happen there, okay? Now obviously, similar with other signaling events, we also need to have a way to return back to the original state. Okay? We can't just keep releasing neurotransmitter and getting rid of vesicles. We also have to make new vesicles, and we have to recycle those proteins. Okay? So what happens is, after the neurotransmitter has been released, we get the opposite process of exocytosis. We get endocytosis, and a new vesicle is formed. Okay? And the new vesicle is then filled once again with a neurotransmitter, and it can go back and be released. Okay? The whole cycle of the exocytotic or endocytotic, depending which part of the cycle we're talking about, vesicle is extremely complicated. It's very tightly regulated. There are many different steps in it. It's a lot of stuff going on there, but it's not really important for understanding what goes on. I'm just going to mention one additional protein, which is known as an abbreviation, as an acronym, NSF. NSF. And NSF is a protein, it's actually an ATPase that untangles the snare complex. So I said that the snare domains are like Velcro, they want to stick together. So we need a fair bit of energy to release the complex so that we can have the vesicle primed as in the beginning. And NSF is an ATPase, it's an ATP consuming enzyme that does exactly that. Takes the whole snare complex and just untangles the snares so that they can be reused. Yes?
Does the proteins attach to the snare, do they attach to each other before the calcium signaling is coming or after?
No, the snare domains only stick together after calcium goes in, okay? They are relatively closely associated, and as I said, there are many other proteins around that regulate it, but no, the actual sticking together occurs when calcium comes in. Right, last thing that I'm going to say is that since this, two last things, okay, because they're quite important for the test. First, well, let's talk about toxins, okay? Since obviously the signaling in the nervous system is very important for life of all animals, okay, at least most of them, many creatures developed toxins, very specific, very toxic chemicals that can target various places in the signaling and thereby kill or incapacitate the animal or human or what have you. The first toxin I want to mention, should have mentioned in the first part of the lecture, is a specific blocker of voltage-dependent sodium channels. So we saw the role of sodium channels, very important for action potential. If you block them, there is no action potential, you can't move, okay, you die. Very simple. The toxin I want you to remember is called tetrodotoxin. Tetrodotoxin, very specific, very potent blocker of voltage-dependent sodium channels. The toxic dose, the LD50, so the dose that kills approximately 50% of humans, is just a few milligrams, 1 or 2 milligrams. Very, very potent toxin. Does anyone know where it's found? Have you heard of the fugu fish? Some of you have? Okay. It's a delicacy, reportedly, in Japan, some kind of fish. Anyway, it contains, in some of its entrails, it contains high concentrations of tetrodotoxin. So if it's not prepared well enough, it kills the person who eats it, okay? I mean, obviously, if it's prepared well enough and they take out all the entrails that contain it, it's fine. But there were quite a lot of deaths caused by that. Okay, so tetrodotoxin, a very potent blocker of voltage-dependent sodium channels. The other two toxins that I want to talk about have a completely different mechanism of effect, and they are produced by bacteria. So they are bacterial toxins, and those two toxins are enzymes. They're huge proteins, about 150 kilodaltons, very big proteins, that have very intricate ways of getting into cells, because they need to get into a cell for them to work. Okay, but I don't really have time to talk about how they get into cells. Very clever, but they do. Once they get into a nerve cell, what they do, as I said, they are enzymes, and specifically they are proteases. So they cut proteins, okay? They degrade proteins. And the proteins that they cut, that they degrade, are the snare proteins. Okay, so those are bacterial toxins get into nerve cells and cut up the snare proteins and thereby disrupt exocytosis of neurotransmitters and therefore stop the transmission of signals in the nervous system. The two toxins that I want you to know are botulinum toxin and tetanus toxin. Botulinum, B-O-T-U-L-I-N-U-M. Botulinum. Who's heard of Botox? Just raise your hand. Okay, most people have. That's botulinum toxin. Okay, Botox, that's the abbreviation, botulinum toxin. Botulinum toxin, if I'm not mistaken, yep. Botulinum toxin breaks down SNAP25. And tetanus toxin breaks down synaptobrevin. Okay, so those two toxins produced by bacteria, they get into our nerve cells by very clever ways, and once there, they disrupt the exocytosis of neurotransmitters and therefore kill us. Botulinum toxin causes a complete paralysis because it targets especially nerves that innervate the muscles, so they stop your muscles from moving. Okay? And that's also the reason why Botox is used in cosmetic surgery or cosmetic medicine, because what you do is you paralyze the muscles that basically hold your skin, and therefore, you remove the wrinkles, because as the muscles can't really move, the skin just kind of lays there and there are no wrinkles there. So that's the reason why Botox is used. Tetanus toxin causes an exact opposite. It doesn't cause paralysis, but it actually causes spasms. Okay, muscle spasms. And the reason is that unlike botulinum toxin, which targets those nerves that innervate muscles, so the excitatory nerves, tetanotoxin targets inhibitory neurons in the central nervous system. So if the release of inhibitory neurotransmitters is blocked in the nervous system by the same mechanism, you get the opposite of paralysis. You get muscle spasms because there's no inhibition of whatever is going on in the nervous system. Yes?
That is a toxin released by also when you get rabies?
No.
No, because you get there's a shot that is called anti-tetanic.
Yeah, but it has nothing to do with rabies. Rabies is actually a viral disease. It's caused by a virus. While tetanus is caused by bacterium, and it's caused by the. But you're right that both of these diseases, when you get them, can be treated by antibodies. So you can get a shot which neutralizes the toxins because they are proteins. So you can have antibodies against those. But rabies is a separate thing.
So you get this. I know that another way being treated by anti-tetanic is when you culture with something that is oxidated.
Yeah.
So you also get the bacteria.
Correct. That's what causes. Well, botulinum is actually. You find it usually in food, so you eat the toxin. With tetanus, you usually get the bacteria in you. They are anaerobic bacteria, but you'll learn a lot about that in later years. Yep?
Did I understand correctly when you said that there's a treatment for rabies?
No, I didn't say that. I said that rabies is a separate thing.
Okay.
Okay.
Yeah, that's what I thought. Thank you.
All right, okay. I'm sorry that we're ending late, but all right.